on diatom's research in different fields of diatom bio-nanotechnology. Dr. Vandana's dedication to scientific inquiry and her pioneering work in diatom nanoengineering and metabolism exemplify her commitment to advancing knowledge and fostering interdisciplinary collaborations. As we gather to exchange ideas and explore new frontiers in science, let us extend a warm welcome to Dr. Vandana Vinayak and embrace the opportunity to learn from her insights and expertise. Thank you. Ma'am, the stage is yours.
Okay.
Thank you so much.
Thank you. Thank you so much, Liquid Trees. Rather, it's my pleasure to give a talk in collaboration with Liquid Trees on this
My
particular international
specialization, as
colloquium
mentioned in
of
the
microalgae and diatom research. Uh, okay, well, it was a little longer. Uh, so let's proceed our talk with. So it's on the diatoms as a future of the global crude oil. Thank you very much. Okay, this all has been said in the two-person and the number of the projects, including the latest currently Indo-French projects, around 2 crores, which is ongoing with us. So let's start with the origin of the diatoms. So diatoms is something not that we have discovered just now or a decade back. It has been 185 million years ago, and they are playing an immense role in representing the climate change through all these era, because not only of their silica walls, as you all know till now that the diatoms are made up of silica walls, the pattern of the fatty acids in the sediments of the diatom, and especially they are having one biomarker which is known as the 24-nor cholesterol marker, which is a biomarker of the diatom oil only. And it contributes to the crude oil deposit since 1839. The research immensely we started on online medias or the offline medias, and the record shows, especially the Oamaru diatom samples, they shows that the diatoms of the Eocene and the Oligocene era, they were distributed to the Oamaru slides. So I have observed some Oamaru slides, and we have published some research publications in the widely also the Oamaru diatoms gifted by someone in England. So I say them that they are the living jewels, because they have such a beautiful architecture. There are more than 20,000 different kinds of the diatoms, all of different shapes and sizes. As you know, there are two orders, pinnales and centrales, and the remarkable nano architecture of each diatom, because of the well arranged pores. The nature has engineered the pores in such a refined and equally arranged manner that even the well-trained engineers, they can't do that. So as you can see, these are the SEM images of the diatoms, and this is the first tool, that is the silica wall of the diatom. So we say them as the nanobots in the glass shells, because they are made up of two cell walls, that is the apitheca and the hypotheca. And because of their nanopores in size, which are ranging from 1 micro to 10 nanometer in size, they have immense properties in the nanobiotechnology also, besides other properties which we will discuss, and mainly of the crude oil. So being rich in the silica, the surface of the diatom, it is rich in many functional groups like the hydroxyl, amino, silanol, and all the different, and also the LCPA group. And all these groups, they are varying in different species of the diatom. So we collected around 12, 13 different genera of the diatom. We cultured them, and we grouped them as per the functional groups which were analyzed via the FTIR techniques. So grouping them as per the amino groups, keto groups, ester groups, and the silanol groups, and the LCPA groups, we found that the different shapes, they contributed to the different group of the different functional groups also. So I can't go much in detail, but yes, you can definitely read this publication of ours. All the slides in this lecture are our own publications, by the way. So like we have grouped them in group 1, which is rich in lipids, polysaccharides, and peptides. Then group 2, for example, group 1 was contributing to Nischia species 2, Gomphonemma parvulum. They were all the pure cultures. And then the group 2A contributed to Melocera variants, Nischia species, Nischia palea, Pinula.
Hello, ma'am. I think your video is paused. You can switch off the camera and switch on again to continue. Ma'am, your video is paused. You can turn off your video and then again turn it on.
Please bear with us for some time. There are some technical glitch. We'll resolve it as soon as possible. Yeah.
Like using different statistical tools, PCA. So this was the assemblage of 12 different diatom species which were cultured in the lab, and then they were grouped based on the presence of their organic compounds, whether they are low or high. For example, we found that the Nischia species 2, Gomphanema parvulum, they fall into category 1A, which had low concentration of the lipids, polysaccharides, and the peptides, whereas the group 1B had Catenula species, Nischia species 4, Salaphora, which had high concentration of the lipids, polysaccharides, and peptide based on the functional groups. Similarly, we had group 2A, which had Malocera varians, Nischia species, Nischia palea, Pinularia saprophylla, Placonis alginensis, which show the presence of inorganic components of high condensed silicates. And similarly, Diodesmus confervaceae and Cyclotella meningiana, it shows the inorganic component of the high concentration of the amorphous silicate. So the FTI proposed model, it showed the presence of different functional groups, which via the surface chemistry, they are distinguishing the diatoms in their different morphology, not by just the microscopy, but also by observing the different functional groups. By using a basic tool of the FTIR, we can have an assess that this diatom belongs to this particular group, and the same study can be done for the different diatoms. So as I said, the diatoms, both living or dead. So for example, if I say these are the dead frustules, and over here you can see this is animated pheodictylum tricornutum. So this is having the chloroplast in between. So they serve as immense, they have immense application. For example, the dead diatom, it serves as a raw material for the silica, the organic silica, which can be used as a vector in the drug delivery systems, as an analytical tool, as a sieve to sieve the adsorbents as the adsorbent material, then the biosensors and the nanomaterials, nanoparticles, and the biological stuff, that is lipids or the chloroplast. They also have their own role. For example, the oil, it is rich in crude oil, and besides the pigments in the diatom, that is the fucosanthin, it is having it is rich in antioxidant, and it is having the nutraceutical properties like the proteins and the phenolic compounds besides the lipids. And the diatoms, they have this unique property that they are monitoring the water quality, because their shape and their quality, or their species, they vary as per the quality of the water. For example, if the water is contaminated, it's having the heavy metals, they show some deformities that I will discuss with you later slides. So what is the problem right now with like we know that datum is so good in producing crude oil, but why? What is the problem? Like the whole globe across, we are facing the increasing oil price, and with the advent of the electric vehicle, why the electric vehicle has finally been impunged into every place. Or after Paris Agreement, you can say since 2015, many countries took a pledge that we need to reduce the GHG as the rise in temperature by plus 1.5 degrees Celsius. So the pledge was to give an era of electric vehicles by 2030. Now this has its own lacuna, which I will discuss. But right now the problem, we have the increasing oil price and the consumption of the current fossil-based products. So we have come up to this diafuel. So it's over here, I somewhere mentioned. So this you can see, this is the patent we got for the, as the diafuel is the diatoms and the fuel is the biofuel. So it's the biofuel from the diatom. So that we got an Indian patent for that. So we say diafuel, that is the biofuel from the diatoms. They have, the potential of making India independent of the imported oil. Why India? Because India is having immense sunlight, and obviously India also has a high requirement next to China, or probably equivalent to China because of the increasing industrialization and the population. So the idea of the diatom solar panel, it's not the solar panel, we all know solar panel is something, a silicon-based body, which is exposed to sun for the electricity. So now the word we have joined over here is the diatom solar panel. It is a living solar panel, which will take the sun's energy to manufacture the oil as well as electricity, which we will discuss. So this may replace the photobioreactors, the current photobioreactors. Why? Because the capital cost is very high. The current cost of those bioreactors is very high because they kill the algal cells to extract the oil. And, killing the alga or the diatom is not the solution, because every time you have to put another inoculum, and the cost of the cultivation increases, and when the cost of cultivation increases, so does the cost of the products extracted from them increases. So the capital cost is very high of the photobioreactors. The productivity is not equivalent, right? And this is a closed system, and in open pond cultures, we are prone to the contaminations. So these struggles are there. So the technical difficulty in sterilizing these photobioreactors is a major hindrance for application of the mass culturing of the algal culture for our desired end products, whether it's the biofuel or other value-added products or VAP, including the pigments or other proteins and carbohydrates. So as I said, these are reservoirs of the crude oil. So these are few pictures of the diatoms via LM light microscopy. You can see these oil globules. These are the diatoms, and these are the different species. And these are the oil globules which are stained via Sudan 4. So this is simple schematic diagram. Like the diatoms, they are just taking sunlight. They are taking the GHG, and these diatoms, they are producing the diafuel, which is good enough to replace the electric vehicles, the ban to the lithium batteries. We don't have reserve for lithium batteries. We all know we don't have reserve for lithium batteries. India doesn't have any reserve for lithium batteries, and the concept of cultivating the solar energy, wind energy, nuclear energy, water energy, since the pledge was taken and the advent started, it was a very slow growth. And many leaders, they have warned that what if this thing fails, like if the backup fails. Right now, we are getting most of our electricity from the coal. Now, switching off, replacing coal with this renewable energy sources, solar, wind, nuclear, all these sources, for the electric vehicles, which would be definitely having the zero gas emission. But what about the battery waste they would be creating? So the battery waste would be one thing, the lithium waste would be one thing, and for some reason, if the backup is not good enough. So now, right now, at this stage, I'll say every house is owning at least 3 or 4 cars, because every individual is working. So the emission of the gases right now, the gasoline gas emission is high, and that's why we have a lot of pollution, especially Delhi. And that's why the liquid trees, I think, is working onto it. Now we are switching off to the electric vehicles, zero gas emission. The source is replaced from coal to the renewable energy sources. What if this fails? What if the backup fails? We don't have anything left. We will have to go back to the coal. And then what will happen? We have to go back to the coal for running or charging our electric vehicles. And that would be a disaster. So yes, geologists definitely have claimed that all the crude oil right now, which we are getting, of which the 30% is claimed by the diatoms, as they are rich in the EPA and the PUFA, or the polyunsaturated fatty acids, and the different species they are counting for the different percentage of these neutral lipids, glycolipids, and the phospholipids. And you all know that the niche is one of the most cosmopolitan diatoms, and it is one of the richest source of the PUFA. Other diatoms, genera like Haslea, Rhizostomia, they synthesize certain highly branched isoprenoids like they are C25, and these all studies one can easily do via GCMS. So they are important sources, in short, of the biofuel or the diafuel. And yes, they have the zero gas emission, zero carbon emission. They have zero carbon emissions, equivalent to the electric vehicle, which we are seeing there. Zero carbon emission. The oil from the diatom is such, it's ready to take for the running your vehicles or the transportation. It's ready to be taken in the domestic purpose. So my discussion would be here, some certain economical strategies to get the oil from the diatoms, whatever your diatoms of interest may be, right? So first we have to work on the media, like we all know we are using different strategies to use the F/2 media, whether it's for the marine or whether it's for the fresh water. But we need to develop strategies to, have economical media, like whatever of using the pre-treated wastewater, because pre-treated wastewater or the wastewater, it is rich in the nitrate, phosphates, and there's a certain trace metal and ions. Besides silica, if is an additional component, for the datum, that needs to be added. And that also can be economically harvested. Because in our lab, what we do is, earlier we used to purchase this sodium, this metasilicates, but lately we have been synthesizing the silicon nanoparticles from the, paddy, rice crops. So the strategy would be not to extract the oil from the diatoms, because once we extract, we are killing the diatoms. It's just a simple process I'll discuss in step by step. The first would be to select certain self-oozing diatoms. Then if we don't have self-oozing diatoms now, since the wall of the diatom is very thick, there was one patent by in 2012 by Phillips and Green, which said a slight pulse of electric current on the microalgal wall, which is having thick microalgal wall, it fractures the cell wall, right? And the nutrients, they come out, whatever the nutrients of interest are, they come out. And when you feed them more fresh nutrient media, the wall heal up. So working on that particular strategy, we developed the dye-sensitized solar cell. We developed the lab-on-chip devices. We developed the microbial fuel cells, right? And we replace the wastewater for not only for remediating the wastewater, but also culturing the diatom economically, and certain economical techniques like the bubble farming for mass culturing the diatoms. And what would be the expected green future when we use all this? So the point, as I said, is milking, right? Okay. As you can see, this particular cartoon over here, the lady is milking the cow, right? So it's like you have the cow, you milk the milk every morning, but you don't kill the cow. So why we kill the diatom? You can milk the oil from the diatom. Why to kill it, right? So now for, just harvesting is a term which is not killing the algal cells, and the extraction is killing the algal cell. Now the extraction, it needs the regular intake of the nutrients, CO2 obviously, and the new algal strain cell harvesting is a little different. And the milking, it's a closed, process where whatever the dead materials would be of the diatoms, it would be used for the new generation of the diatoms to take in as organic matter for their growth, whether it's the nitrate, phosphates, or the silicates. So we have to develop some techniques, or we have to optimize those techniques, whether it's the using certain solvents or the centrifugation or via doping, or whether it's the mechanical pressure, or whether it's PEF or the natural oozing properties, right? So there was this datum which is known as the Diadesmis confervaceae. Over here you can see this datum. It was having the property of the self-oozing. The datum, there was no external pressure applied, nothing was done. So the datum on the 15th day, it was all full of the oil globules, and it finally, the wall, that the wall of the datum, it is very thick, and it needs the pressure equivalent to 750 micronewtons is required to burst the wall so that the oil comes out. Now you can see over here the diatoms are there, and the oil globules, they are coming out without killing the cells. So if you feed it more nutrients, so it is surviving. It was one of the very best diatoms on which I have studied. And you can see the oil on the 31st day, all the oil is coming, and now the diatoms are becoming dead. So the point was to monitor the growth and the pattern of oozing. So in this particular, we found that 14.6% of the oil was coming out via self-oozing property. Simultaneously, our collaborators in France, they were working on this species known as the Terpinsone massica, which worked on just a mechanical pressure. It was given on the just a cover slip on pressing the means a shear pressure of pressing the slide in the diatoms. So the oil was coming out in these Terpinsone massica. So besides the oil, crude oil, which can be used directly, the biofuel, it is the esters, that is the fatty acid methyl ester, which will work with the methanol and produce the glycerols and the methyl ester. Now, step by step, I would be discussing some techniques. So over here, you can see the atomic force microscopies of some diatoms of the control as well as the test samples. In these test samples, this is a 3D view and this is a planar view. So over here, this is a control and this is a 3D of the test. And similarly, this is a control, this is a 3D of the test. So whatever you see over these are the pores of the diatoms, and they are getting impregnated with something. Now this something is, we have encapsulated the gold nanoparticle, and plus this gold nanoparticle has impregnated into the pores of the diatom via some peptide bonds. We have induced the peptides like biotin and tryptophan, and after doping them with biotin and tryptophan, which was a chemical process, we have encapsulated with the gold nanoparticles. Now on exposing them to the sunlight, right, there was a thermoplasmonic effect on the nanoparticles, which allowed the oil to come out. So this is in detail. You can study the paper. I'm just giving you a little idea of what's going on. Similarly, we worked on doping the diatoms with the magnetite nanoparticle. This is the HR10 of the diatoms doped with the Fe2O3, showing the lattice structure that the diatoms were doped with the Fe2O3 or the magnetite. So once the diatoms were doped with the magnetite nanoparticles, now putting a magnet outside the flask, it attracted the diatoms to collect at one particular site, because since they are doped with the magnetite. Now whatever the oil which you have harvested by any technique, whether it's your self-oozing diatom or whether you have used any other technique, harvesting technique, to let the diatom ooze the oil. So the oil get collected on the top clear. There is no artifact of either any biological material, because the diatoms are there, the pure culture, and they get attracted towards the magnet because they are doped with the magnetite. So that was one study which was published. Then we had also studied how these are all the economical studies, in the oil spillage cases, where how you absorb the oil, right? So we developed this technique where we had doped the this thing, Lufa cylindrica. This is Lufa cylindrica is a vegetable. So its fibers we have taken. We have doped with one of the linkers, that is PDMS. PDMS is an adsorbent, right? So we have used simple PDMS So over here, you can see the simple PDMS. It's a hydrophobic substance. So it is having the properties of absorbing the oil, right? So this is a contact angle over here. You can see the contact angle of the PDMS alone was 112 over here, and it got reduced to a certain degree. And then we had used the PDMS, which was the loofah cylindrical sponge, which was coated with TiO2 and PDMS, and the oil. This red color is of the Sudan 4 to to let us visualize the oil. So this was completely absorbed, and the contact angle was decreased. So over here, this bar chart, you can see that the alone PDMS, this is the oil absorbing efficiency of the PDMS alone. PDMS is a hydrophobic substance, is 19%. PDMS PDMS loofah was 18%. TiO2 coated PDMS was 25%, and TiO2 coated PDMS and loofah was 38%. So these all were the economical technique compared to the genetic engineering technique, which we all know. It enhances the lipid. So this is like you have the datum, you do some metabolic or genetic engineering. So metabolic is still cheaper because you can play with the nutrient factors, whether it's a nitrate or phosphate. You can give stress to the diatoms, because under stress their whole Kelvin Benson cycle, it shifts, and they start producing more of their value added products, whether it's the lipids or whether it's their other value added components like the fucoxanthin and the pigments. So this increases the accumulation of this value added products once it is in stress. So this putting the diatoms in stress via making a deficit of any chemical like nitrogen, phosphate, silicate, or any other hormone like melatonin, etc., or even high light. It increases, it puts the diatom under stress, and it start increasing the oil content increasing. But even if the oil is increasing in it, you don't need to extract it. You need to harvest it. Then you should have some techniques to harvest it. So that would be a very simpler technique compared to the other genetic engineering techniques or the CRISPR techniques, which increase the, like it has been studied in PT, that is the fudictylamine trichromatum, that there was a 45-fold increase in the tag through disruption of a particular gene, which is the UDP glucose pyrophosphorylase gene. Obviously, it increases the power of the diatom to harness the biofuel. But yes, it is not economical. You need the gene gun. You need all those genetic engineering setup. So if you see very simple, this is a very simple structure or a diagram or schematic diagram, which we aim for, that this is our diatom solar panel. This is just a tank, right? But I mean, this is a rather closed tank, but it can be a visible tank, which probably a better diagram I'll show you further. So it can be on your rooftop, something on your rooftop. The diatoms being cultured, they are taking the sunlight, right? And some technique is there that they will be either they are the self-oozing properties diatom, or there's some technique from where they will be oozing the oil content, and that then there would be some sieving thing where the upper surface of the diatom or the some adsorbent like PDMS or loofah, which will absorb the oil, and the oil then get collected to your tap, and you're just opening it and you're feeding your bottom tank, and you're then feeding your domestic purposes, or whether it's the transportation purposes. So all this is written over here. I need not read that. Now that was the diatom solar panel, which is something, the diatoms on the rooftop, and then you are milking them, right? Some techniques are there. So why using the photobioreactor, or what would be your tank like? Like I have just shown a tank, but how this tank can be, how can you model this tank? So recently, since 2019, one of our collaborators, we proposed this bubble farming techniques. It's like the bubble wraps which we are using in every packaging products. So these packaging products, they are having this bubble wrap with air in them. So we plan to test the growth of the diatoms in these bubble wraps, but then we need to test that which quality of the bubble wrap would probably promote the closed system and the economical way of growing the diatoms. So for this, we tested various mechanisms. First was the regular open flask model, that is the open culturing or the open pond culture, right? Then was the first flask which was sealed. These were our trial experiments, which were then later materialized. So then there was a flask having the diatom culture, which was sealed with the natural density polypropylene. Then we had sealed with the plug was sealed with the high density polypropylene. Then we had sealed the mouth with the low density polypropylene. So all this thing was done and the growth was monitored, all the parameters was monitored. So over here, I'm just showing the diaphyl content over here. As you can see, in this particular, when it was an open culture flask, we had a mixed diatom growth and the production of the diaphyl was lowest. So then over here, when it was sealed with the natural density polypropylene, again, it has the low diaphyl, and in HDP, the growth was less. But in the low density polypropylene, it has a high content of the diaphyl. So bubble farming was using, definitely this is because of the emerging high cost of the biofuel from the diatom, which is approximately $3.36 per liter. So this bubble farming by using different plastic bubble wraps, because it doesn't need, and in my lab, the cultures which I have kept of these bubble wraps, they were maybe 3 years old. I have not feeded them, no nutrient feeding again. There is no contamination, there is no water loss. So there is no post-nutrient uptake, no water loss. For this experiment, by the way, was for 40 days, right? And the maximum diaphyl produced was 37%, and the lipid was 35 microgram per ml on the 40th day. So this was published in Bioresource Technology, by the way. So these were the physical parameters, and very briefly I'm mentioning there was the conductivity and the pH compared to the control of the LDPE, and the water retention properties of the LDP. Over here you can see that so this is the open container, and this is the water content, and this is the bubble wrap. There is no water loss compared to the control, which is having the water loss. So these are some setups which, again, this was published in Science of CETA, Sustainable Energy and Technology, and this was in RSC. So you can see the different techniques. We have also used the the glucose bottles used in the hospitals, the bubble wraps which comes in the packing material. So all these things different. Even we are planning to use the catheter bags also, if that works for us or not. So coming to the economical source of the nutrients, so using the wastewater for microalgal culture is something not new. I'm not saying something new. It was developed in 1950. Since then it is being, but yes, definitely if you want to use it for the edible stuff, like whether it's the tablets or the pharmaceutical or nutraceutical, you need a pre-treatment. But for the, and also you need to see the pH and everything. You need to pre-treat it, even if it is for the oil or the biofuel. So the wastewater, it is entering the algal cultivation, and from the algal cultivation, it can give you not only the clean water, the wastewater gets remediated using the algal cultivation, whether it's a diatom or any other alga which you have tested it, right? It gives you, it remediates the water, it gives you pigment, it gives you biomass, it gives you biofuel. It can be used in the novel technology known as the microbial fuel cell, which where it gives also the electricity. And this biofuel, it can be used as an alternate for what's going on right now. So because it is having the zero footprint, as I said, biomass as a food, and therapeutics rich in the DHA and the ESPA, and in the medicinal properties also for the fishes. It's better to live in the clean water. So I have not added over here one slide, but I want to share with you. NASA recently has used this technique of this is a recent study, but ours had been a little previous study. They had used this I will not say bubble polybags, yeah, but these are the polybags only. They have said they have used their polybags in the water, and in the water are the alga, microalga. So they are taking there's a omega plastic bags. You can study it. So they are taking the contents from the wastewater for the nutrient for the algal cultivation or the algal biomass increase, and plus the alga is not facing the crisis in the waste water, something like this. So we have tested various different kind of the pollutants for the datum growth, like we tested in 2019. This was published in the again in I think Wiley. Yeah. So this is effect of wastewater pollutant. We tested different pollutant like paint. We also tested the growth of the isonia, the water cyanide, sewage water, detergent, then the beta drugs like the beta propanol hydrochloride, even the crude oil, how they are being degraded by the diatoms, and then the silica salts. So these are the different studies, and in short, I will tell you that the diatom responded well to the crude sewage, and at 40% and 60% dilution, and that was the best study. For example, this graph was showed the best study when they were cultured in the 40% and 60% dilution, and there was elevation was 10 times compared to the control sample which was devoid of any sewage water, which was in the F/2 media. Besides the other drugs, and even the crude oil to certain extent shows the good growth, and higher contents of the silica salts were taken. You all know that we use metasilicates for the diatom growth, but the concentration above what we use was used, so the diatom they couldn't sustain the high silica contents also. Now, the metal pollutants, as I said, they bring stress to the diatoms. So this was another study where we found that the diatoms which were collected from one of the sites in, that is Saraswati Dham in Kurukshetra, Pewa district. So this particular, we were sampling the diatoms from different water bodies of the Haryana, and that study was done almost for 4 years. And for this particular site, it was found that the datum, one of the datum was showing a distinct shape, right? So this datum was identified as Gomphonemus pseudo-auger, and it showed the morphology. It was different from its normal morphology. For example, the normal morphology is this one, the first one. It showed this small, the deformation over here, you can see. But on comparing the oil bodies in the control sample and the diatoms which were deformed, it was seen that the oil content or the lipid content in these deformed diatoms or the deformed Gomphonemma, it was more compared to that in the control. So these the heavy metals which were discovered in this water body was a lead and the selenium, and it finally showed that the stress, the heavy metal stress, it deformed the morphology of the datum and also put it under stress in their Kelvin-Benson cycle, and they were producing the more of the lipids. So this is just a simple diagram, schematic, where we have shown that the pre-treatment is very essential, whether it's via the ultrasonication. The ultrasonication, it is of the wastewater, as well as the diatoms, they are disrupting when they are in the wastewater. So how this would be collected, this is just a brief study, and I think I should go a little faster. So I was talking about the live diatoms and how the morphology get changed, and they start under stress, they're producing more of the diatoms, but also the dead diatom, as I said, they are having these pores, and these pores, they increase the surface area, and they have their properties to absorb the heavy metals. So the metal organic framework of different diatomaceous earth or the diatomite, it has shown the removal efficiency of the lead by 96.45% of all the different kind of waste industry we have, like the food industry, laundry, iron, and so on, thermal. Like TT, that is the furictalum trichromatum, it shows ability to absorb the cadmium metal ions, and not only the cadmium but the chromium metal ions also we are doing in our lab. So they show good absorption of the lead, cadmium, copper via some phytochelation effects. Similarly, the Nischia, which is the most cosmopolitan diatom, it tends to show absorption for the zinc, copper, cobalt, manganese via different van der Waals and the pi-pi interaction, and even the non-covalent interactions. So absorption of the cadmium was also seen in Thalassiosira vesplogi. So you can see that these diatoms, they are smart nano containers for absorbing the heavy metal pollutants. So over here, I'm just schematically showing these are the dead frustules of the diatoms, and over here how you can dope with different nanoparticles also, because they are rich in selenol groups, as I said. So you have to make a stability of the surface of the diatom, so that whatever the adsorbent or whatever the pollutant is there, it easily get absorbed onto its surface. So that you have to test on, so that you get the clean water. And as I said in my previous slide, that the wastewater, if it is rich in the pollutants, especially the heavy metal, it will deform the diatoms. But if you dope with the nanoparticles, the deformities caused by the heavy metals, it will deform it, the lipid content increase, and once then via thermoplasmic effect, the nanoparticles, they will slightly fracture the cell wall, and the oil content or the other biocompounds, they easily come from the diatoms. So case of dead diatoms and the live diatoms. Now coming on to the dye-sensitized solar cell. So this, we all know what a simple fuel cell is regarding. We all studied the simple electrochemistry in our classes, lower classes. We have one anode, we have one cathode, and some electrolyte in between, and the potential is generated, right? So this dye-sensitized solar cell, it is having a technique which, as you see, the word dye is there. So we have again we have the cathode, we have the anode. So these, we are taking the FTO, fluorine tin doped coated glass slides, and they are coated with the anatase TiO2 dioxide and the dye. The dye has to be photosensitive, because on exposure to sun or to the light, they are taking the photon of the light, which is absorbed onto this TiO2 nanoparticles, and then electrolyte is there in between, which can be any electrolyte. For example, it can be your simple Lugol's iodine also. It can be wastewater also, by the way. So I have not tried it, but yes, I have studied some papers onto it. So this is a cathode, which is your semi-transparent something like platinum, right? So on exposing it to the sunlight or the artificial light or the incident solar radiation, as I said, so there's a electron is produced because of the electrolyte in between, and when the electron flows, the current is produced. Now how you will see that we have used this technique for our biofuel. So this is via metabolically engineering the Ti2 nanoparticles onto the Nischia palea, one of the diatoms. So that was via two-way feeding. So we had the SiOH and TiO2 into our diatom culture simultaneously using the syringe pumps. The syringe pump was working manually in our case earlier, but not later. But then we did the experiment in IIT Bombay. So, and the pH was maintained. So this was all optimized first, and this feeding was done for 8 hours, and then it was left in dark, and then finally exposed to light. And then these metabolically engineered TiO2 nanoparticles, they were characterized via the different state-of-art instrument like the AFM. So over here you can see these are the empty pores of the diatoms and how they are getting doped with the TiO2 nanoparticles. So this is a 3D image. And then this is, you can see over here, this is a small FTO plate. The cathode and anode is prepared. They are clipped, and they are doped with the anatase. And we have this metabolically engineered diatom. Over here, I'll go back. So this is the FTO glass. This is the FTO glass. This is a control. This is a test. So this was a dye plus anatase TiO2 using the conventional dye sensitized solar cell. So this is the
Ex- Ma'am, your video is paused. Can you please turn off your camera and turn it on again? You
from
can
which
continue,
slide?
ma'am.
This slide? Where was I?
Ma'am, slide is visible. In case you have network issues, can you turn off your video so that we can continue? And
I
we are
don't
running
see
short
network
of
issue.
time. Yeah, we are
I'm
running short of time, so we can just wind up soon. Yeah.
so okay. Okay, I think I discussed this. I don't know whether you heard it or not.
Yes.
I was talking about the metabolically engineered diatoms and how they are used in the dye-sensitized solar cell, and how the oil was recovered, and how the oil was recovered from these diatoms, and on feeding more of the nutrient media, the diatoms, they were alive, and this was the oil recovered on the different days of the datum, and it was on the 18th day upon 1%. In this, this is a 1-inch FTO plate. So then we designed another strategy. Why to, because the earlier strategy of metabolically engineered was a very cumbersome process. So we now use the simply doping the diatom with the silver nanoparticles via the two different peptides. There were two different peptides were used. One was the biotin tryptophan, and another was the allelitic tryptophan peptides. And we stabilized the silver nanoparticles on the diatom. This was the cyclotelamine Gianna. So we found that the biotin tryptophan, it enabled the more stability for the silver nanoparticle. And this doping, after this doping, they were again used for the dye-sensitized solar cell. And compared to the earlier studies where we had used the metabolically engineered diatom via two-way culture technique, in which the efficiency of the diatom dye-sensitized solar cell was 9.45, we found that this simple technique, a facile technique of the DSSC, diatom dye-sensitized solar cell technique with TiO2 and silver nanoparticle peptide embedded diatom, it shows the efficiency of 8.5. It was little less, but it was more faster compared to the earlier technique. So this is a schematic again showing how the on exposure to the light, the oil will be coming out because they are doped with the silver nanoparticles. So this was a lab on chip device which we engineered at IIT Bombay under our project with IIT Bombay. So via this is as you are saying, this is you're running short of time. I have to skip or go fast. So this is a BioMEMS device where we have fabricated a small lab on chip on a 2-inch wafer, silicon wafer. So we tested various designs, and we found that this circular design was the best via the various masks which we used. So this lab on chip, which was fixed with the PZT, which is ceramic, something like ceramic, which was fixed at its back, and it was converted to the oscilloscope and the function generator to let the diatoms in these micro wells, which are having these micro pillars, these micro pillars, to resonate at a particular frequency. The frequency was set as per the calculating the size of the diatom, the shear strain it can tolerate. So all this was calculated, and then the how much resonance energy needs to be given, it was decided. So then it was observed via the different microscopic studies, and how the oil it oozed out without killing. So this was a resonating device which was made for the biofuel production. Then later on, another comparative study which was published in Bioresource Technology, we took the Pinularia saprophylla and we compared the MEMS device with the centrifugation device and the pulse electric field. So we compare because the wall, the problem is the wall of the diatom, silica wall. So over here you can see the hydrophobic wall of the diatom in all these three cases, and how in these three cases we will be getting the lipids from the different techniques, whether it's the resonating device or the centrifugation or the pulse electric field. So it was found that the centrifugal force of 1110 xg for 15 minutes, it allowed 3.39 percentage of the lipid, but there was 2.5% cell destruction, right? Now, in the case of the PEF, there was 21.19% of the cell death with 2.86 of the lipid production for 10 microseconds at 110 kilovolts. And for the case of the resonating or the MEMS device, we had 14.15 of the cell death and 4.4 of the percent of the lipid which was recovered in 20 minutes. So this was a better device compared to the other, and it was economical because then later on we developed the diatom solar panel as a benchtop model, which is already filed for patent. Right. So next, this is a microbial fuel cell technique. We all know what a microbial fuel cell is. We have anode, we have cathode, we have a proton exchange membrane in between, which allows the protons to pass through it. So one is your anode, other is your cathode, and at the anode we have your wastewater, and we have some microbes over here which are oxidizing the organic matter over here. So once they are oxidation will occur. So over here the oxidation and the reduction occur simultaneously, and once the oxidation and reduction occur, which flows, and once it flows means the current is generated. So this is very brief discussion. So I will try to add up over here that if over here the purpose of the cathode is to something which is a oxygen, generator, something which will give you oxygen. So it can be air only, or it can be water with regular sparging, or it can be a chemical, which nowadays people are using generally the chemicals by which some reaction will take place, and the reduction and the oxygen would be generated, which the hydrogen ion which is passing from anode to the cathode via this proton exchange membrane will be converting into water, and the process of electron movement will take place, right? So but over here what we have done, we have placed the alga over here, we have placed the diatoms over here, which by the way will produce oxygen via the photosynthesis. So this is a free production of the oxygen, so no chemicals are required, no regular sparging is required. So you're using the you can use the wastewater, you can use the organic matter, you can use a dead diatom over here, because the dead diatom will absorb the heavy metals in the wastewater. So we tested, I'll skip on to this, the different alga were used, but the diatom was never used. So we, for the first time in the microbial fuel cell, we use the diatoms, and at the anode, as I said, there should be some microbe, but these microbes, they should be electrogenic. So if it is not electrogenic, the current is not going to produce because electron is not being released. So for the first time, we use the diatoms, like using the diatom at the cathode is easy because it's a photogenic, but then again, certain parameters which you have to adjust. But for the first time, we use the diatom at the anode, and we proved that the diatoms are electrogenic also via the mechanism of the NOx sign which are being generated. Now, to shortage of the time, I will not discuss much in detail how the NOx signs are generated via NADPH, the urea cycle and all this stuff in the night. So we didn't use micro, we used the wastewater, we used the diatoms, and the current was generated, and the diatoms, they generated the oil. Over here you can see the actual process where we connected a series of these microbial fuel cell, and the simple LED bulb was enlightened. And you can see this is different polarization data, and the pigment data, concentration of the pigment, and the concentration of the pigment in the anode side. And this is the polarization curve, or the efficiency of your cell. So this was the 12.62 milliwatt per meter square, and the Coulombic efficiency was 22.95. The datum cells, they showed 64.28% of the increased lipid production on the 15th day, beside the production of the pigments. Increased pigment was found on 15th day, 4.890. So what we learned rightly is that we need to fracture and let the diatoms heal, right? So via the different, we can optimize the technique via PEF, nanoparticles, mechanical pressure, whether it's the resonance, or electrolysis, whether it's the MFC or NADSCC. So this is a slide just showing that the diatom also are good absorber for the hydrogen being produced in the microbial fuel cell. So I'll skip on to it. So now next thing which we learned is that there are different techniques, economical techniques of harvesting, not only the diatom but other alga also, beside the costly photobioreactors which we are using. You can use the simple bubble wraps for economical harvesting. You can use the photosynthetic microbial fuel cell for harvesting the diatoms and other alga. So the proposed solution is, in short, the roof tank model of the diatom solar panel via any of these technique which may suit economical. So this is a schematic diagram of, for the inhabitants in this room, they are exhaling the CO2, and this CO2 is going to another chamber or another room via this poured walls, and the CO2 is taken by the diatoms, which are pushed to the top layer, where we have this micro resonating pillars, right? And on the top, we have certain layers of adsorbent material, like let's say PDMS loofah, which will absorb the oil, and that oil will come out in the form of diafuel. And this is all thermal insulated, and the transparent glass sheet for the sunlight to come and avoid the evaporation of the water also. Now, how to maintain the temperature? This was in collaboration with some other people in California. Well, this was suggestion. We have not yet worked onto it. This is on the large scale, pilot scale, where we can use the dynamic walls for our climate, because India is a tropical country, high temperature is there, 41, 45. So you need to maintain a dynamic wall where there's a cool air drawn in and the warm air is drawn outside. So other green future content via biofuel is you're taking the wind energy, solar energy for the electric vehicle. There's accumulation of the lithium batteries, which I told you. Accumulation of the
Excuse me,
coal
ma'am.
energy.
Sorry to interrupt.
Yeah.
Extremely sorry for the interruption, ma'am. Actually, we have ran out of time. It's nearly 15 minutes. Sorry.
Yeah, done.
Yeah. Can we just go for the next thing? Yeah.
Yeah. Okay. Should I end?
Yeah.
Uh,
Yeah, ma'am.
should I
Sorry to
finally
interrupt, but yes.
officially end or just abruptly end?
No, ma'am. You can just continue for a minute and then
Sure.
continue.
Yeah, yeah,
Yeah.
yeah, yeah. So that the novelty is to use diapole instead of the electric vehicle. So the atoms, they are the small milking cow. Harvest, don't extract them, don't kill them, and they are your future in moons and the Mars mission. Whether it's in the for the food for the astronaut or the fuel for the astronauts, or even for the terraformation of the Mars planet, it is not like you will go and plant a tree and the tree will grow in the Mars. You have to go from the microorganism, and the atoms are the best substitute because they are the 40%, they are primary producer, 25% they are absorbing the GHG gas emissions. So the applications in drowning deaths, which I have done 4,000, 5,000 cases. Recently a patent was there in Germany, fingerprint powder, some media collection was there. And I thank my Indo-French Sripra project and my PhD students, Ankesh, Vanna Srotia, Priyanka Gurpreet, and Urvashi Soni, who is now also a PhD student. And I will invite you to join the international conference on September 12th and 13th, which is on the green technologies, innovation, and startup ventures. Please do join. Thank you very much.
Thank you so much, ma'am. It was a really overwhelming session, and you have a lot more to offer for us. And definitely, you have done a lot of research work right from the uh, history of the diatoms to the newest technology, nanotechnology, using all the possible economic techniques. And it is really appreciable work, and you're doing very good research, and we are definitely would like to keep the session again for you, and then we can address some issues. But right now, due to some time constraint, we cannot move to the question session. But I would welcome you to answer the questions in the chat box if you're happy with it. Yeah.
Yeah, yeah.
Otherwise, we can share the email address to the attendees, and then they can continue with it, ma'am.
Yeah, yeah.
Yeah. Thank you so much, ma'am. Thank you for the session. Yeah.
Thank you so much. Can
Yeah.
I leave?
Yes, ma'am. If you would like to answer the questions in the chat, you can. Otherwise, we will pose the
There
questions
is
to you in the mail.
there is only appreciation. There is no question.
Questions and answer session in the tab Q&A tab is there.
Which
Just yeah, beside the chat option on
no, no,
top.
chat I have
Yeah.
seen, but chat only I can see. Where are the questions?
Q&A separate tab is there, ma'am, beside to the chat option.
Okay.
Yeah. Yeah, thank you so much, ma'am. I would like to give it to the next
Yeah,
for the next theme.
yeah,
Yeah,
yes.
yeah. Please continue. Thank you.
Good afternoon, everyone. So my name is Arshita Nigam. I'm a microalgae cultivation scientist at Liquid Trees. Now we have our next title, theme like exploring microalgae and diatoms, innovation and strategies of carbon capture and sustainability. So as per this theme, we have a esteemed speaker and well-renowned scientist from IRI, Dr. Sunil Pabbi here. So Dr. Pabbi is have done his PhD as well as MSc honors from the Punjab University, and he was professionally employed at ICAR IRI New Delhi.